Riccardo, you give the start?
I suppose that you start and
Okay.
You introduce and sorry, I'm waiting for
perfect,
you. So.
Perfect. Okay. So good afternoon, everybody. Thank you all for sharing this event with you and for attending to this event. A special thank is to the Interdepartmental Center ECLT and the IUSS Pavia, that especially the IUSS Pavia who hosted this event, and a special thank also to all the speakers of this event. They are people very busy and outstanding, but they are happy to share the discussion on this issue. This is a special event in the sense that is a joint class of 3 courses, 2 courses of the Department of Law. One is on Law, Science and New Technologies, is the course where I teach. The other one is on Industrial Law, and the teacher is the Professor Marina Tavassi. There is also a 3rd course that is a course of the use, where I teach as well, that is on information technologies and law. So the focus today is on synthetic biology and the patentability of inventions in the field of synthetic biology. And this is a very engaging and challenging issue, but we have to consider that the background of this problem of patenting in synthetic biology is wider, because synthetic biology has a lot of implications from an ethical point of view, social point of view, legal point of view, even moral and religious point of view. Patenting is part of the problem, is a very important part, because there are great economic interests on synthetic biology application, but it is only one part. This is the only thing I want to say in order to stress the importance of this focus, and at the same time, the partial approach that the patents can offer. But now I have the pleasure to give the floor to the Professor Riccardo Pietrabissa, who is the rector of the IUSS Pavia, that for his opening address to this event. And I thank him because I know that he's very, very busy and he's in another meeting at the same time now. Thank you, Riccardo. You have the floor.
Thank you very much. I welcome each of you to this afternoon meeting that is, in my personal interest, very, very important. Synthetic biology and patentability. This is a core problem, I think, that we should face rapidly in the next years, and the idea to have a dialogue, let me say, a comparison between experiences and problems between the US and the EU is very important. We know that the disciplines in IP differ from USA and the EU. For some topics, and so these differences may be very useful also to understand and promote something that allow to you to manage the global, because this is the real problem. I like to thank very much Amedeo Santosuosso, that is a new good friend of mine. We are working on different topics, and this is for me an extraordinary good experience. I have an engineer, a biomedical engineer, is a lawyer, and the idea to put together different disciplines to face a real world, let me say, real world is very important for each of us and for the future generation. I'm rector since less than 2 years. I'm the COVID rector of IUSS Pavia, that is an institute of advanced studies that face its job not on disciplines but on facing themes, challenges. We like to put together people that have different background, because the problem normally is not only one face, but manifold faces. And the topic of synthetic biology and IP is one of these. Synthetic biology is a sort of pretty new word. I check on some definition of system biology. I was interested in that, and I found on Nature, on the journal Nature, a very synthetic definition of synthetic biology, which is designing, construction of new biological parts, or the idea to construct biological parts, devices and system, and redesign of existing natural biological system for useful purpose. This useful purpose is something that, in my mind, can link to the patent, because patent is an exclusive right. So the idea to exclude someone that is referring to a technical solution of a problem. So problem and useful purposes are the two links in my idea of this point. But many questions may arise that has the ethical problems that in general face on patent. You know that there are ideas, ideology that are against the possibility to patent something that is useful, for instance, for health. There is the complex idea which had the right, who had the right to apply for a patent that involved biological matter, and which is the concept of invention applies to biology. So many of these topics in my mind are as no solution, and I hope that you can help us in identifying possible solution, and in particular that this comparison between different experiences in US and in EU may help us in find a possible framework to discussion on that. So let me thank again Amedeo. Amedeo is a very important man in this period for me, because I'm understanding a lot of new things. This is important because as a professor, I like to learn something new to teach every day. And so to put together technology in intelligent artificials and biology and law and economics is the real world. So let me thank him. Let me thank Andrew Torrance from MIT, Boston, Ilaria Delisa, is a lawyer in Munich, Marina Tavassi, dear colleague of University of Pavia, and Silvia Garagna, that I remember we already met many years ago with our good friend Carlo Redi, and Lorenzo Pasotti. Both of them came from differential background because are bioinformatics and biologists. So have a good afternoon. I'm very happy that to host this important seminar, and have a good discussion.
Thank you, Riccardo. Thank you, Riccardo, for your address. And now we can move to the first part, because the first part of this meeting is a scientific part. And this is the result of the experience we had in the research center ECLT with Silvia Garagna, Carlo Alberto Redi, and many other colleagues. Our starting idea was, before talking about the law of something, the law of genetics, the law of biology, the law of neuroscience, and so on, we lawyers have to learn some about the scientific side of the story. Otherwise, we can give a lot of talks, but without any meaning. And so this is the reason why I have the pleasure to give the floor to Silvia Garagna, who is responsible of the Laboratory of Development Biology in Pavia, at the University of Pavia. And her field of study is reproductive biology in mammalians. And she is my boss now, because she is the president of the center ECLT. I am simply the scientific responsible of the center, but it is always a pleasure to work with Silvia, and I ask her to introduce from a scientific point of view this event with an introduction on the frontiers in biology today. Thank you, Silvia.
Okay, thanks, Amedeo. I'm sharing the screen. I have some slides to show you. So first of all, also my thanks to Professor Pietrabissa, to all the participants, to the speakers for accepting this invitation, and to Amedeo in my double activity today as a speaker and as a president of the European Center for Law Science and New Technologies, mainly for your tireless efforts in promoting opportunities like this one that allow a continuous, really useful dialogue between science and law. And thanks also for inviting me as a biologist to share with you all what I consider to be the frontiers in biology at the beginning of this millennium. So today I will show you the impact of biotechnologies on cells, tissues, organs, even organisms, producing living forms that do not exist or develop in nature. Also inspired by the imagination of some writers and film directors, I want to envision with you future development and use of the results of the present day research. I think that the most well-known engineered cell is the minimal cell. Minimal cell was originated 5 years ago by the group of Craig Venter. He was able to synthesize a genome with 473 key genes to introduce the genome into a genome-free living cells. These cells are bacteria. They behave correctly. The only thing at that time was that they were not able to divide producing cells from the same size. And this is very important because this means that the most important event in life is reproduction. They were not able, these cells were not able to reproduce correctly. 5 years later, in these days, the same group of scientists published a new paper where just adding 7 genes, 5 of which of unknown function, make these cells to divide perfectly dividing symmetrically, giving rise to 2 cells of the same size. So now these scientists have generated cells that perfectly mimic natural bacteria. By engineering the genome of these cells, we can imagine to shift the single cell life of this organism into group of cells, a super organism that can be induced to perform some activity, like, for example, as it was said when the paper was published, to clean oceans and so on. But not only single cell living organism can be manipulated, also cells from complex organisms like vertebrates, say amphibia or mammals. And just published is this beautiful paper in which Michael Levin and co-worker were able to produce, starting from stem cells, mobile self-organized clusters of cells taken from xenopus frog embryos. This is the movie summarizing the experiments. They were taking, these are the embryos at the very beginning of their development. They were cleaned, and only part of the embryo where the most important stem cells are located were dissected and then left to grow for 4 days. After 4 days, they formed spheres, and these spheres were able to move because you see, they move very well in very several directions because they differentiated small hairs, and the beating of these small hairs let these xenobots to move. They sense the environment, being able of changing direction. They can record the experience by emitting light when exposed to specific light. They become green, but also they move particles. You see, they clean the space where they are from very small particles, but very important, they are able to self-repair when injured. They are not able to replicate. They are not able to produce other organisms like they are. So they can be considered only somatic cells, but induced to perform some activities. Stem cells obtained, for example, for mammals, can be differentiated into small tissues or organs. There are many studies in this field generating miniaturized, simplified version of 3D tissues and organs from rats, from mice, from other animals, but even using human stem cells. And this is an example of hair follicles produced from stem cells, and these are hairs that are coming from the follicles. The problem of this type of organoids is their 3D structure. They are very much disorganized, so they end differentiation, but they are disorganized. And now engineer like Lorenzo or Professor Pietrabissa developed what they call organs on chips, where chips are used to control the flow of gas, of media, of growing factors, thus obtaining tissues and organs with the correct architecture. And not only these correct tissues with the correct architecture can be used to model human disease or in toxicology studies or in drug development, but even, and more, this is an important field in this type of research, in regenerative medicine. And just a few months ago, in this paper, the authors show that human cerebral organoids were transplanted into the brain of mice, and these organoids were able to extend projection and develop the good connection with the cells of the mouse brain. So we can imagine that if we will be able in the future to scale up these organs, they can be used for transplantation, avoiding the use of explanted organs from a donor, from donors. Also, these chips have been improved to grow not only one, but up to 10 different tissues, which are connected by a flow of circulating medium or a surrogate blood. So, and these complicated chips called body-on-chips can mimic complex physiological events. We can imagine in the future chips where all the tissues and organs that are present in an organism may be physiologically connected and integrated. Why not hierarchically governed by a brain inside the chip? Among these chips, placenta is one of the tissue that is studied. There is a prototype of placenta on chip. Researchers are studying the chips for producing artificial 3D human placenta. There are some results growing lambs and mice. The first paper was published 4 years ago, and you can see here premature lambs that were grown for 28 days outside the female body. And these lambs were grown achieving the same differentiation in terms of physiological activities, weight, and morphological differentiation as the lambs that were developing inside the female body. This was really an important paper, but I think a turn in these studies is determined by the publication of this paper just published on mice. Mice were developed in these small bottles, fetus, until day 11 of gestation out of 19. The novelty of this paper is the time at which the fetuses have been explanted from the uterus, exactly at the time when the embryos implant, 1 day later. So from the beginning of gestation, now this artificial system is able to grow the fetus for a time that is more than half of the gestation time. But embryos that are usually generated using sperm and eggs can be generated also from somatic cells. This has been done with the cloning experiments, but also in recent years, starting 3-4 years ago, using somatic cells, stem cells, mouse stem cells, researchers were able to produce embryos. And the same results were achieved, again, just published papers by 2 different independent groups using somatic cells, somatic stem cells, either derived from blastocysts or from somatic cells that have been reprogrammed into pluripotent stem cells, aggregate these cells and produce an embryo that developed to the stage, the blastocyst stage, that is exactly the stage when the embryo attached to the uterus. So we can produce in vitro embryos, grow them until the time which they are meant to attach to the uterus, but with the last experiment made in using the mouse, we can grow a fetus from the time of attachment to more than the middle of gestation. So we can imagine in the future to obtain new individuals without eggs and sperm and without the female body. Embryos, as I said, can be obtained by cloning. The first cloned mammals were Dolly, the sheep, and Cumulina, the mouse Cumulina, and since then, many other organisms have been, mammals mainly, have been cloned, but also birds and frogs and others. There is a very interesting research project led by Professor George Church, Harvard University, who wants, by using cloning, to insert the genome, genes of mammoth into the genome of elephant. So manipulate somatic cells of elephant, introducing mammoth genes, and then using cloning to obtain embryos. So we can imagine in the future to have this mixed genome that can be very useful to understand from the point of view of scientists, how genes work, how genes help an organism to adapt in the environment, but also why not thinking, we can think to mix genome and maybe generate new species. How can we call this new form of living systems? I looked for 15 days after Amedeo invitation, the right word. And at the end, I thought that bioids may be the right word or a proposed word to indicate this not natural living systems. The system may be integrated into a body, integrated into an artificial network like the body on chip, so why not think of new types of robots, allowing the somatic generation of individuals, generate a new form of lives, maybe new species, even change the natural history of a living species, and much more of this, changing the natural history of living species, including our the CRISPR-Cas method of manipulating the genome has been employed to manipulate human embryos without much success. There are really very few papers on this topic, but 3 years ago, Yang Kui, a Chinese scientist, manipulated embryos, trying to, with the CRISPR-Cas method, trying to introduce mutations in this gene, CCR5, to confer resistance to the HIV infection. There were 3 newborn. 2 were studied for looking for this mutation, and sadly, no one of the 2, they were 2 female twins, had this mutation. So the technique didn't work, but the new generation were mutated, and we don't know what will be the future in terms of development, of growing of these babies. He was jailed, went back to China, okay? But anyway, this possibilities of manipulating living cells, the genome, organism, tissues, pose many, many different challenges, as Amedeo and Professor Pietrabissa said at the beginning of this lesson. They are ethical, economical. We can think of to have restriction to individual freedom, but also the free will I may want to decide to have my child outside my body. We have to start again to talk about eugenetics. But also, why not think of a more inclusive society, future inclusive society. So what else? Thank you very much for your patience and patience.
Well, thank you, Silvia, for this very, very interesting presentation. A little bit shocking, but the ce n'est qu'un début. And now I leave the floor to Lorenzo Pasotti, that, as I said, is the real specialist specifically on synthetic biology. He has worked with Professor Magni, and they are very well known in Italy and outside for their activity. And so, Lorenzo, I suggested you to use the style of the TED Talks that are around 15 minutes or a little bit more. But okay, we have the pleasure to have you with us today.
Okay, thank you very much. Can you hear me and you see the screen, right?
Yeah.
Okay. So thank you for the introduction and for the invitation to this event. So in this presentation, I will try to give some overview of the state of the art of synthetic biology for non-biologists like me. So after a brief overview, I will show some products of synthetic biology. So I will try to define what are the real products that we aim to obtain with this discipline. And I will end with some examples of achievements that have been done in the last few years to give a more practical understanding of what can be done and what cannot be done yet, for example. Okay, so I start with a definition. As you said before, there is no universal definition. So I normally give mine that try to better describe the field with some customized words, but essentially it is a very young field that aims to mix the capabilities of life science and engineering to construct novel biological functions. And the way to do this is, can be done in two ways. The first one is the hardest way. So if we want to construct something, we can start from scratch. And if we start from scratch, it is very difficult in biology because we need to design and construct an entire genome. So we need to know how to build a new organism, and we need to assemble it practically, okay? And this is unfeasible right now, because even if there have been efforts to construct organisms in the lab, so the synthetic cells that we were talking about a few minutes ago, we still do not know how to design the cell, okay? So we can copy something from nature, but designing from scratch is very, very difficult. And for this reason, probably the most productive branch of synthetic biology is this one, that aims to rely on naturally occurring organisms and to design supplementary functions in order to make them show some unnatural capabilities. And I will focus on this branch in this presentation to show some state of the art. Before doing this, a little bit of more definition from mine. So what is new from disciplines that are well known, like genetic engineering? So the aim is not so different with synthetic biology. So the aim is to construct novel biological functions. So to modify organisms to make something new, something useful, okay? The real difference between the two disciplines is the complexity that can be reached. Because what I normally say in explaining this is that genetic engineering is not really engineering, because it doesn't include a real engineering approach in handling the design, the complexity, and so on. And analogously, synthetic biology is not really biology, because it has a much stronger influence from the engineering world in terms of design and handling the complexity of everything, okay? So if we want to construct novel biological functions in to reach a real complexity, we need to handle the complexity of biology, which is nowadays huge, okay? And in synthetic biology, we want to do this by developing tools to help this process. So in order to facilitate the design of new organisms. Designing new organisms, what does it mean? It means that we have to write something like this. So we need to write a repetitive code of ATCG as the DNA is. And after we know how to write it, we can put everything together. And all of this can be inserted into an organism, like a cell, a bacterium, a mammalian cell, or so on, in order to let the cell express unnatural functions. So we can reprogram the cells with our writing of the alphabet to make something new, for example, sense some inputs from the environment, process this information, and respond with the production of specific molecules and so on. And this is quite a good picture that recapitulates quite well our thinking as engineers in the lab in order to build up and design novel biological systems. So we start from inputs, we need to process them, and we need to give some outputs, okay? The same writing of this DNA code is usually seen in parallel as computer engineering. So with computers, we do normally more or less the same things. So we program like softwares and we let our computer make something new or increasingly complex. That's good. And it is exactly what we want to do, starting from DNA, programming it, and making our machine do something even more complex than the naturally occurring ones. And in this case, the machine is, of course, the cell. It can be reprogrammed with these tools. Okay. Just to mention what are the main aspects that, in my opinion, distinguish the synthetic biology field from others, are essentially these three. So they all are from engineering concepts. So the first one is to have a collection of components. So if we have a list of good parts, the engineers can put them together like bricks to build up something. Okay, this is the concept of having individual modules that are taken and assembled together. Then we need some design concept. So we need rules to combine everything. And in this case, engineers can have the probably major contribution, because most of the work of the engineers is based on design. So we normally use softwares, computer-aided design tools, and so on to try to design the best systems we can do. And finally, of course, we after the selection of components, after the design, we need to construct practically all the things. And in this case, we have the bottleneck in biology, that is the construction of DNA molecules that need to be assembled or synthesized in the lab. So in this case, the contribution of automation and laboratory robots, but also new kits that can facilitate all this process are very desirable. So these are like catching pictures that are normally present in synthetic biology publications to illustrate this concept. So cells are seen as a chassis that can be engineered with specific circuits, like in electronics. Then we have toolkits of components from which we can take the best parts that we want to assemble everything. And the assembly process can be done like we do with Lego bricks, actually. It is not true, like it is not as simple as I said in the lab, but the concept is to facilitate the assembly process as much as possible, and that's it. So with all of this, what can we build? So what are the main products of synthetic biology? Usually the products are considered the organisms. So the product of synthetic biology is usually the organism. And these organisms can be used for different applications. Here I just divided the application into two classes that are not exhaustive of everything that can be done, but it is a quite important separation of the two things. So first of all, the cells can be used as factories. So we can use the cells as factories for the production of biomolecules, or we can use the cells as interacting components with their environment. What does it mean? So factories for the production of biomolecules means that we can take the cells, let them grow in an industrial plant, and as an output, we can get the molecule of interest, like a fuel or a drug or a specific material and so on. So that's the a classic biorefinery concept. Okay. Then what about the systems that can interact with their environment? There are some fascinating examples. So for example, cells like bacterial cells can be used to be inserted in our body, like probiotic cells. So bacteria that are able to grow in our body, colonize some specific districts, and do something for us. For example, pathogen killing in case of infections, and reporting some disease state, so for like living diagnostics. Okay. Or analogously, we can do this with the plants, so in the environment, in order to get some toxin detection or toxin degradation for bioremediation in the environment. And the main distinction between these two things is this one. So on the left, we have cells that are usually confined into a tank or an industrial plant, okay? While on the right, all the examples need to have the cells free in their environment, okay? They need more closer interactions with our body, the land, the leaves, and what we want as an environment. So on the class of examples in the right, pose a very interesting and critical aspect in terms of biosafety, because we need the bugs or the cells or whatever we use as an organism to be engineered. We need them to be safe, or we need them to be biocontained in some way. Okay, this is a quite important concept in this case. Examples of the first class of cells are, for example, microbes like bacteria or yeasts that can be used to do a lot of things. So cells can be used as factories with success. They have already been done. It's not very new. So some of these molecules, many of these are already seen on in the market. Okay, so they're not completely new molecules. So it is not a matter of novelty of the molecule to be produced, but it is a matter of the way we want to produce it with the specific cell that has been engineered with some new pathway that is very efficient. It overcomes the bottlenecks of the previous ones. And as a main point, in synthetic biology, we can add very complex pathways that actually lead us to use very cheap substrates to produce everything. So in our production process, we can actually start from waste materials. So there have been wrappers starting from solid biomass, from wastewater, and even from toxic gases that can be reconverted as a feedstock for the bacteria or the yeast to produce something useful like these molecules, okay? And these molecules can be fuels, chemicals, and materials, as I said before. In the second examples, I just put some high-impact studies in the field of medicine, in which we have here some clinical trials that are ongoing, so a very advanced phase, in which we have probiotic bacteria that have been engineered to do something in the human body. First example is to cure some metabolic disease. So, for example, here, the first trial is on PKU, that is a metabolic disorder, in which the patient accumulates a toxic level of a specific molecule introduced with diet, and the bacteria that can be as well introduced in the diet as a pill can consume this toxic molecule for the patient. So in order to cure this disease. Another example here is a bacterium that can be injected into solid tumors. So we can trigger an anti-tumoral pathway starting from engineered microbes to cure actually cancer cells, okay? So in this case, it is also worth mentioning the biosafety aspect, in which these bacteria have been engineered also not to proliferate in human body districts that are not the tumor. So this is quite an elegant example of the topic of biosafety, in which we want the cell to do its job, but we want it to do in the place that we want. So not everywhere. And if it goes everywhere, it dies or it is not able to proliferate. And that is a good example. As for the environment, it is quite analogous. And here we have two examples from two industries that have developed some organisms in order to carry out nitrogen fixation that help plants to absorb nitrogen. So we can have a reduction of nitrogen fertilizers for agriculture. That's it. Okay, so actually the examples are finished. Here I just make a brief summary of everything. So first of all, of our capabilities. Our capabilities are improving. So the good news is that in the last 20 years, biology has indeed become easier to engineer, also for non-specialist people, so non-biologists like me. So this is a very good aspect. But in order to exploit the full potential of synthetic biology, we need to improve the points in which synthetic biology is stronger. So all the engineering aspects that can handle a very wide complexity in the design processes. So we need to expand the toolkit. So we need more bricks specific probably for more unknown organisms, the so-called non-model organisms that are not actually the ones very studied in the lab, but they are very suitable for many applications. We need to overcome the bottleneck of DNA construction that is a slow process, and we can improve it in terms of time and cost also. And we need to improve the success. That is the most difficult point, probably as an engineer, I can say. We need to improve the success in achieving an engineered organism that work well for us. So when we design, construct, and build, it is not always occurring that the organism is working well, because sometimes the design fails. Okay, so we need to improve this success. In terms of de novo designed organisms, we need to expand a lot our abilities to do this. As we saw in the last presentation, we have projects about minimal cells, minimal genomes, and also feasibility studies in which people have tried with success to synthesize in the lab entire genomes. That's very good as from the technical point of view, but in terms of application, we don't have examples yet, and it is worth mentioning the order of magnitude of the complexity of writing a genome from scratch compared to writing an individual function alone. Okay, and as a last slide, for the available products of synthetic biology, we have some good news from the last 10 years, actually, in which synthetic biology has delivered real products that are in their later stages of development, and some of them are already used in market. So we have more fuels, disease treatment, food additives, and so on. Everything as a warning, always thinking about the safety aspects that always need to be addressed when doing the design process of genetically modified organisms. We saw some examples, there are more, but I just put this sentence as a final one, just to make sure that every design has its phase in it. And that's it. So I thank you very much for your attention. I thank you for the invitation, and all the lab people for their continuous work in the field in very interesting applications, both in some of the areas that I've talked about today. Okay.
Well, thank you very much, Lorenzo, for this extremely clear presentation that gives us the possibility to move on the legal side, having a solid, good ground where to work on. And if it is possible for you to stay with us also during the time of legal presentation, it would be nice, because if there is something that is involuntary, inaccurate, you can intervene and saying, okay, this is not exactly true from the strictly scientific point of view. It would be very helpful for us.
Yeah, we'll see.
Thank you so much, Lorenzo. And now I have the pleasure to introduce Ilaria Dell'Issa. Ilaria Dell'Issa is PhD in industrial law. She got her PhD at the University of Hanover, but she graduated first in the University of Milan, and then she had a LLM at Cambridge, UK, and then she had a period of time at Berkeley University in the United States, And so she is Italian because she was born in Italy, but she's quite international. Nowadays, she is a brilliant lawyer in Munich. And so it is a real pleasure to give the floor to Ilaria Delisa, that is the author of this fantastic book on specifically on the patentability of synthetic biology inventions. So, Ilaria, you have the floor.
Thank you very much, Amedeo, for the very kind words of introduction. And also thank you very much to Professor Garagna and Professor Pasotti for the technical introduction, because this is really a great springboard for what we're going to be seeing now, which is more the legal aspect of it. So really the patentability of synthetic biology. And as you will see, some of the topics that they touched upon, we're also going to touch upon them, but more, as I said, from a legal perspective. Now, before we start, the first question is always, what is a patent? And as we probably already know, it's an exclusive right, as we said before, to an invention if you can provide a product or a process that has a new way of doing something or offers a new technical solution to a problem. And this somehow already plans out what are the requirements if you want to have a patent. First of all, you need an invention. And this basically means you cannot patent something that is a discovery, something that is a scientific theory, or that it's a law or a product of nature, as they say in the US. Basically, the idea behind it is that you cannot appropriate for yourself something like the law of gravity or general relativity theory. Those are things that are supposed to be for the use of mankind, and you should not have an exclusive right on them. And as we will see, this is a topic that is very relevant with biology in general, because as the Rector already said, it's also a question of appropriation and what we feel is appropriate to cover through a patent. Other patent requirements are novelty, inventiveness, and industrial application. And another requirement that is usually very pivotal in biotechnologies is exceptions from patentability, and especially the morality clause. So you might have an invention that it's new, it's industrial, it's inventive, but is it moral? Is it something that the patent system thinks is moral enough to patent? And as you can imagine, in the biotech field, that's always an open question. Now, what we're going to basically go through now are some patent applications and patent examinations. There are no court decisions so far strictly on synthetic biology, just because, as we heard, it's a very new scientific field, and that's the reason why cases on this have not reached the court yet. But they might in the future, and already patent offices have started to deal with this topic. And what you can tell by the patent offices dealing with it, and also from the scientific introduction that we just had, is that synthetic biology is definitely a new technology. And because it is a new technology and something also very technical, you can expect that it's going to push even further the boundaries of the patent system. It's going to ask questions which the patent system right now is not able to answer. And this is where this presentation tries to outline what are the main possible issues. As I said, no courts decided on it, but this is what we expect might happen. Now, as good lawyers, the first question is always, which norms apply? Like, what are we talking about? First of all, TRIPS will be applicable. This is an international treaty coming from the WTO, so the World Trade Organization, and this applies to patents in all fields of technology. So from car technology to biotech, this covers everything. Something similar is also the European Patent Convention. It also applies to all sorts of technology, so also to synthetic biology. And what it's important here to notice is that it's a European Patent Convention, yes, but it's not a EU, so it's not like a European Union convention, which means that states like the United Kingdom and Norway are signatories to this convention, but they're, as you know, not part of the European Union. So let's try to keep those two separate. We also have, in addition, a purely European Union norm, and this is the last one, the EU Biotech Directive. And this directive considers only biotech inventions. So it really focuses on this. And what it does is determining the criteria for biotech patents, and it does in a way that somehow it favors their patentability. By favoring, I mean that it offers some criteria that make it a little bit easier, or at least explain some of the main issues that you might have with biotech patents. It doesn't mean, however, at the end that if you just apply for whatever patent in biotech and you're going to get it just because of this norm. It just means that it makes it slightly easier because it clarifies a number of issues. Now, having said this, and this is the general overview, what does it mean on a practical sense? And here, I hope that I don't say something wrong. The scientists in the group might correct me anytime. There is a field of synthetic biology, which is called xenobiology. And amongst others, this is aimed at creating an expanded genetic alphabet or creating DNA with a new backbone. What this means is basically, you all know you have a basis in DNA, A, T, C, Gs. And what scientists were able to do was add 2 new bases, X and Y, and also change the backbone of DNA. So as you know, DNA, the D in DNA that we see here stands for deoxyribonucleic acid. What they did was exchange that and create DNA with another type of acid, so the thionucleic acid. So it became TNA. Now, this is all very scientific, but what is the approach here? The issue is, once you do this, not only you have something that is not available in nature, but with the expanded genetic alphabet, so with the new basis, you have something that has problems replicating. And what you have, if you get the new backbone, so this new part, you have something that is invisible to natural biological system in many cases, with the situation that you find yourself really at the boundaries of what we understand as biotechnology. Now, let's see this one in practice. So we said, this is the scientific part. We have seen the theory. How does it apply? So if you have something like this, something that is not available in nature, that has problems with replicating, and it's invisible to biological system, does it get the application of the Biotech Directive? And remember, the Biotech Directive, if you want to have a patent, is really good for you because it allows you more patentability leeway. So if you apply for a patent, you do want to apply this. However, the Biotech Directive also has a definition in it that says it applies only to a biological material. And in this field, biological material is defined based on these 3 criteria. So genetic information that is reproducing and is reproducing in a biological system. Now, if we compare the 2, you can easily tell that this definition does not sit well with xenobiology. Now, this all seems very abstract still. The thing is, it has a very practical repercussions, both legal and for those who try to seek to patent something in the synthetic biology field. If you see it from a legal perspective, you can say, well, you know what, you can still apply the Biotech Directive. It's still a biotechnology. You make just an extensive interpretation. And last but not least, we are lawyers, we are legal scientists, we're not scientific scientists. So why should we use a scientific criteria that we don't understand? Let's just apply it. It should not be a problem. And if society at large sees this as a biotechnology, it should also not be a problem. On the other hand, you really have a definition that goes against what we just discussed. That there are some parts of that definition of xenobiology and biological material that actually clash. And you can also say, and this is an argument that is going to come more and more with synthetic biology, that is it really biological if biological systems cannot see it? Is it biological if it's so different from what we already have in nature? Like, can you make that argument? And as I said, the consequences of it are really remarkable. Well, if you can't apply the Biotech Directive because you choose to go down this road, what is the consequence? That you only apply the European Patent Convention and TRIPS. So far, so good. But it means as well that you cannot apply the directive. And the directive is a European norm. And as Professor Tavassi and Professor Santosuosso can easily tell you, this triggers the referral mechanism from national judges to the European Court of Justice. So if a national judge has a question on a European norm, they can trigger this mechanism and somehow also have a clear opinion from the European Court of Justice, which then is somehow unified for entire Europe. What you have, if you can't apply this norm of European origin, is that every judge in the EU can actually choose a different approach. You might have, especially on morality questions, a decision in Malta, which is very different maybe from something you will have in Lithuania, just because those topics are so different. And of course, getting away from the opinion of the European Court of Justice might prove to be complicated and problematic. Last but not least, you have an issue. If you want to apply for a patent, as we said, you're much better off if you can apply through the Biotech Directive. So if you apply for a patent, more difficult for you as well. Now that we've given this framework, let's just look specifically at what are the advantages of the Biotech Directive and really in practice what this means for us. As we said before, if you want to have a patent, the first thing you need to prove is that you have an invention. Now, at European level, at the EPC, you say, if you have an invention, as long as you don't have, for example, a discovery as such. So far, so good. The Biotech Directive, however, helps you very much if you are in the synthetic biology field, because it says you have an invention if you produce biological material through a technical process, even if that same biological material previously existed in nature. And this is a major thing because, as you can imagine from what we just heard from Professor Garagna and Professor Pasotti, synthetic biology will be done in the lab. And technical process simply means something that can only be done by humans and not by nature. So by using this, you're most likely to have an invention independently of whether something previously occurred in nature. And this is a major advantage, as we said. Now, I touch upon something that Professor Garagna previously showed, so the minimal genome. As we saw, this was the genome, and they removed the dispensable parts, so those purple things, purple genes, and they tried to patent the yellow part, so the minimal genome. This was subject to a patent application in Europe and in the US, exactly the same content of the application. Now, do you think it's going to be considered an invention in both? Or is it going to be a difference in Europe and the US? Well, it is a main difference because in Europe, they thought it was an invention. They didn't even ask the question. They said, you know what, you did it through a technical procedure, even if it existed in nature before, would not have been a problem. And it was so far that people didn't even ask the question if it was an invention or not. Somehow in the European system, this issue got a little bit sidestepped. It doesn't mean that at the end the patent was granted, because it wasn't, but they chose other criteria to reject the same patent. Whereas the US, and I'm talking about exactly the same patent application, said, no way, this is a product of nature, it's not an invention. Because as Professor Torrance will probably show later, they said there are no marked differences compared to what already exists in nature. And based on a long-standing case law in the US, they use this aspect to say they are a product of nature and you cannot consider an invention. So you have exactly the same application. One place is an invention, one place is not. And you might think, okay, this is just an exception, probably elsewhere is different. Actually not. There is another example, and again, I touch on what Professor Garagna said beforehand, in the de-extinction field. Now, there are talks about bringing back to life, as we said, both the mammoth and also the Neanderthal. Also, Professor Church discussed that. Now, we would end up not having an identical copy of what previously existed in nature, but you can easily imagine it's difficult to say that it's an invention if you're talking about something like this. Now, this field is still not being used to patent applications. So this, what I'm saying, is still theoretical. But what people expected is that in Europe, it's probably going to be considered an invention. Because as we said before, even if it exists in nature, but if you did it through a technical procedure, you have an invention. And let's remember, if you don't have an identical copy, it's even more difficult to say, even in Europe, that you're having a discovery. In the US, it's also likely that this product of nature topic will be raised, even though, of course, at the same time, you have to think, what is nature? If something is extinct for years, for thousands of years, can you still consider it as part of nature? Or is it a bit too far-fetched at that point? And this also becomes relevant because you were talking before about the Dolly the sheep case, the first mammal that was cloned. Well, in the US, there was a case about it. And even though it was clearly something that was done in the lab, it was considered to be non-patent eligible because it was too similar to what already existed in nature. It did not have marked differences. So you see, you have two countries, two systems where the approach is very, very different. Now, I said in Europe, the extension will be considered an invention. It doesn't mean, however, that it will be heading for a patent. And in Europe, this is most likely not only on the novelty and inventiveness requirement, but also because of morality issues. Now, I don't need to explain to you, but you already probably feel it. If you think about issuing patents on recreating the Neanderthal, recreating the mammoth, you already can feel that legal and morality questions are going to be there. Now, both TRIPS and the EPC have a norm about this, and they say you cannot have a patent if an invention is against order public and morality. Now, the European Union has issued the Biotech Directive, and in this setting, it kind of repeated that. So it said, okay, it should not be against morality and order public, but they also went further and gave 4 examples of something that they consider to be immoral and therefore unpatentable. One is cloning of human beings, processes for modifying the germline of human beings, uses of human embryos for industrial and commercial purposes, and last, processing for modifying the genetic identity of animals, which are likely to cause them suffering without any substantial medical benefit, either to man or to the animal itself. Now, while this is not an exhaustive list, you can already tell that it really has a focus of human dignity and animal dignity. And this, of course, is going to be fundamental when you think about the extinction work or work that somehow goes towards humans and higher life forms. And this is somehow something that also brings us to another question. What is morality exactly for synthetic biology? Is it a discomfort with the technology? So I don't like synthetic biology. I think they shouldn't be doing it. Is this counting against issuing a patent or this? Or it's really just legal criteria that we have to focus on? Is it the legal criteria that has relevance here? And yes, in general, legal criteria are the one you should check. And this is why we're going to take those legal criteria and try somehow to apply them, for example, to the mammoth. Now, the EPC, you see here, probably will argue, we'll have to discuss the order public and morality question. But definitely, these questions will arise under the Biotech Directive. Not only because we have an alteration of the genetic identity of an animal, but because we might have suffering of this animal. I mean, not only are we bringing back to life an animal, but we bring it back in an environment which is not his own. He might be the only exemplary of his species. So maybe in a world where he grows alone, definitely also psychological aspect of it. And last but not least, we have to prove the substantial medical benefit. Saying, you know, it's scientific curiosity, or we want to put it in a zoo, it's definitely not going to be enough for this. So you see, while in Europe, it might be considered an invention, you might still not be able to get a patent if it's considered immoral. What I just said is basically multiplied by 10 if you think about the Neanderthal. Why is that? Because whether you consider the Neanderthal as human, quasi-human, you still are dealing with something where in the Biotech Directive, you would need to discuss question of human dignity, human embryos, cloning of human beings, alteration of the germline. And you see, those are going to be definitely tough points when it comes to patentability. And even under the EPC, so the standard of morality and order public, the issue is still also quite interesting, because in the past, the EPOs, the European Patent Office, has discussed the case of human-animal chimeras. And it said, while instinctively it is distasteful, and you might even feel it's immoral, there is no consensus on its exclusion from patentability. So you will see the techniques as well as the specific of the public feeling, of the general feeling, and the legal stance that will be applicable at that time will definitely make this a very interesting discussion. And honestly, one that will probably be the central point in the patentability. Now, what does this mean? Like, what can we take out of this, since we said it's still quite a general topic and right now no specific patents were analyzed on this? Well, we have to admit that this stage with this technology is not going to be enough. We need to focus on legally relevant arguments, and definitely public perception will have an impact, but it's the legal arguments that will count. Right now, the legal arguments tell us that it's probably not going to be a major issue, morality right now, because as we saw, there is a focus on higher life forms. So right now we are dealing with microorganisms. So it's probably it's going to be difficult to have morality questions right now. However, it is already very clear that once we transition to animals, human life forms, embryonic materials, these questions are going to be raised and are going to be pivotal. And I hope this presentation just gave you a little bit of an idea of how many questions does actually synthetic biology pose. Because as any new technology, whether you define it as something groundbreaking or an extension of something that happened before, you still see that the scientific development pushes the boundary of the patent system and forces it to reconsider its definitions and its approach to things. So I hope this was interesting. I hope you enjoyed it. And this was it from my side. Thank you very much.
Thank you so much, Ilaria. Very fascinating presentation and very good connection between the scientific side of the issue and the legal implications. Now we have to move on the other side. We have to cross the Atlantic Ocean, and we find Professor Andrew Torrance, who is also a mix of a lawyer and a biologist. He's a professor of law, but he has a PhD in Harvard on genetics, and now he's working at Broad Institute in MIT, and he's currently a visiting professor in my class. I have this pleasure. I met Andrew some years ago at the annual meetings of the Rettore Institute in California, and it was a pleasure to see him and all the other colleagues. So, Andrew, it is up to you. You now have a clear point from the scientific point of view, a very clear point of the situation on the legal side with an approach European, as you can see, but informed internationally. What about you? You have the floor.
I don't think I have anything to add, Amedeo. I think everybody has said so much wisdom. I'm not sure I can add.
Bye-bye.
But I will, don't worry.
Okay.
I wanted to thank you, Professor Santosuosso, very, very much for inviting me to take part in this wonderful event. I've learned a tremendous amount from the previous speakers, and thank you very much, both from the legal and the scientific side of things. I want to point out that one of the gifts that I received by participating was my colleague, Dr. Dellissa's wonderful book. So all of you should get this book. It's very illuminating on the subject. I'm going to share my screen, hopefully successfully, and make it full screen. So I'm hoping that you see something that says synthetic biology at the top.
Yeah.
Okay, good. Excellent, excellent. So what I'm going to talk about is going to overlap a little bit with what some of the speakers have presented already, but I'm going to present, I think, a few new ideas as well that I hope will be complements to what other people have talked about. I'm going to give a very brief introduction, not so much to the science, because I think that's been wonderfully done already, but a little bit to what I see as the origins of synthetic biology and the synthetic biological legal issues in North America, mostly in North America. And I'm going to trace some of the origins back even before the phrase synthetic biology existed. I'm going to talk about the patenting of genes in North America, specifically synthetic biology origin genes, because as my colleague Dr. Dell'Issa has already pointed out, synthetic biology presents some new features to the patent law landscape, which are very interesting. And the courts in North America, just as in the patent systems in Europe, have had trouble grappling with some of these issues. And I want to suggest what I think might be the future of this from the North American perspective. I'm also going to talk about alternatives to patenting. And the reason that I want to talk about that is that synthetic biology, of all of the fields of biology, has been most involved with trying to establish new norms, norms that sometimes avoid patents rather than use patents. So I'm going to talk a little bit about some organizations like the International Genetically Engineered Machines Olympics, the Biobricks Foundation, and the Registry of Standard Biological Parts. And I'm going to talk about that as a way to compare and contrast patenting versus open source genetics. And then I'm going to talk a little bit about the new frontiers of synthetic biology. And it's wonderful that Dr. dell'Issa has brought up the mammoth and de-extinction, because I'm going to share a little experience that I've had thinking about how one might approach the use of synthetic biology to bring back a different organism. And I won't spoil the surprise, but so I want to start a fairly long time ago with an African-American woman named Henrietta Lacks, who was very ill, and she went to Johns Hopkins University Hospital. And while she was there, the doctors discovered that she had very, very interesting tumor cells. And unfortunately, Henrietta Lacks died, but a doctor, Dr. George Otto Gay, harvested some of the cells from her tumor and kept them alive. And in fact, they've been kept alive till this day. And there are now enough of these cells that you could make something like 100 blue whales worth of these HeLa cells. HeLa cells refer to Henrietta Lacks. Now, this did not implicate patent issues per se, but it did get the legal thinkers in the United States thinking about ownership of parts of bodies, ownership of cells, and eventually ownership of the genes that underlie the cells. Now, if you go forward in time a little bit to 1990, there's another patient named John Moore. He had a kind of leukemia, a kind of cancer of the blood, and he was treated at the University of California, Los Angeles Medical Center. He had tissue samples removed, and again, a doctor, Dr. David W. Goldie, actually cultured some cell lines, saved some cells from the leukemia, and he applied for a patent on these cells. The cells were called unique T lymphocyte line and products derived therefrom. That's the name of the patent. And he actually commercialized these cells and sold them and made a little bit of money, but he also got a US patent. And the patient, John Moore, found out about this, and he was not happy because his perspective was, these were part of him. These were from his body. How could somebody else own a part of his body? Now, the court, the Supreme Court of California, eventually decided this issue. And it decided that there was a breach of the ethical duty of the physician, of the doctor, to inform the patient that he was taking cells from the patient's body. But there was actually no theft, the court found. And in fact, Dr. Goldie was able to keep his patent. But this also raised a lot of issues that started to percolate through the American legal system about ownership of biological materials, and ownership especially of biological materials which have to do with human bodies. So let me skip now, or jump now, to genes. 20% of human genes appear to have been mentioned somewhere in a patent, in a US patent. And there's a survey about this by Jensen and Murray back in 2005. Now, that doesn't mean that the patents covered the genes. All it means is that the patents mentioned human genes. there was a lot of outrage in the early 2000s about the whole idea of patenting genes, of patenting DNA. And here's just one example of an editorial in the newspaper. To the person in the street, the grant of a patent covering all potential uses of these genes raises the visceral fear of corporate interests claiming ownership over our very bodies. In addition, Mr. Jurassic Park, Michael Crichton, he wrote an editorial in the New York Times, which is a very influential newspaper in the US. And he said, you or someone you love may die because of a gene patent. Gene patents are now used to halt research, prevent medical testing, and keep vital information from you and your doctor. And although this is a very extreme statement, one thing that I will say about Michael Crichton is that he does have a medical degree. Now, obviously, he went in a different direction, didn't become a doctor. Eventually, he became a screenwriter and a novel writer. But a lot of people were thinking about ownership of pieces of DNA, even back in the 2000s. When you look back over the way that we think about genes, I think that you can trace the origin of the unease that we have about ownership of DNA. More than 100 years ago, Mendel, Darwin, Weismann, and a number of other people, they imagined that there was some sort of unit of heredity, but they didn't know what it was. They really had no idea what it was. They could only see its effects when they bred organisms like pea plants in the case of Mendel, or pigeons in the case of Darwin. Eventually, this unit of heredity was given a name by Johansen, and it was called the gene, and the name gene stuck. In the 1920s, Morgan, who was a geneticist at Columbia University in New York City, he started to imagine that genes must be some sort of particle, some physical thing that if you had a microscope that was powerful enough, you might be able to see, and that the particles were passed from parent to child. So this idea of a particle changed in the 1950s when Watson and Crick were able to decipher the structure of DNA, the A's, G's, T's, and C's that Dr. dell'Università was talking about. And so this particle, it began to be imagined as having a sort of a structure within it that was made of A's, G's, T's, and C's. Now, this idea of specific subparts of the particle eventually began to be imagined as information. And it's obvious why, because the A's, G's, T's, and C's produce things. They have instructions that produce particular proteins. So back in the 60s and the 70s, genes started to be thought of more as information than as particles. And eventually Crick, the same Crick that uncovered the structure of DNA, Crick and Bonner suggested that it's not just any kind of information, it's actually like a computer program. It's like an algorithm. Now, more recently, in the last 15 years or so, synthetic biologists have started to think of genes as parts, as Lego parts, and they call these biobricks. But they imagine that you could take these parts and stick them together or pull them apart and make new things. So I think it's useful thinking about the history of the concept of DNA and genes, because we've now arrived at a time when we think about these things as either information or computer programs or as Lego parts. Now, the US system has actually grappled with the patentability of DNA. And out of this comes a very interesting conclusion with respect to synthetic biology. But let's start back in 1980 with a very famous, at least in the US system, a very famous case called Diamond versus Chakrabarty. There was a scientist named Anandra Chakrabarty. He's currently a professor at the University of Illinois at Chicago. And I had the great pleasure of meeting him about 8 years ago. He's just a wonderful, wonderful man. He loves patents. I don't think that that's a good or a bad thing, but he loves patents. There's no greater passion in his life than patents. And people were asking him questions when we met him about what he thought about the ethics of patents. And he didn't really have views about the ethics of patents. He just thought patents were great. And his answer was, well, I've got 15 patents this year. So he's very enthusiastic about patents. He came up with a particular kind of bacteria, which had been genetically engineered to essentially eat oil. And his idea was that you could take this bacteria and you could put it onto an oil spill in the ocean. It would eat through all of the oil, and then when it ran out of food, it would die. So it would sort of eliminate itself from the environment. Now, it didn't work as well as he envisioned it, but he did apply for a patent, and his application eventually went all the way up to the Supreme Court. And in a very famous line in American patent law, the Supreme Court said that you could patent, quote, anything under the sun that is made by man. Now, I'm pretty sure, I can't prove it, but I'm pretty sure that that also includes made by woman. But the Supreme Court was very explicit that if it was made by man, it could be patented. That's a joke, by the way. I'm certain that regardless of your gender, you can patent genetically modified organisms in the US. Out of this case, the biotech industry in the US really started to flourish. Many companies arose like Amgen, Genzyme, Genentech, and these companies became quite lucrative places to invest. A lot of capital went into them. And part of what attracted the capital was the patents that they held. And so they tried to file as many patents as they could on new types of DNA. The Human Genome Initiative sped this process up to the point where by the end of 2008, 2009, pardon me, not 2008, by the end of 1998, 1999, every biotech company in the United States and many around the world were filing hundreds or thousands of applications on specific human genes that they had identified and sequenced. It became what I've called a gene rush, sort of like a gold rush, but for genes. Meanwhile, in Canada, the Supreme Court of Canada had to deal with a similar issue to Diamond versus Chakrabarty, but this had to do with a Harvard mouse that could get cancer because it had been engineered to develop tumors. It was used as a cancer model. I feel very badly for the mouse. In any case, in Canada, the Supreme Court was asked the question, could you patent a mouse that had been genetically modified? And the Supreme Court in 2002 said, no, you could not. And the reason was that a mouse is a higher form of life, and you cannot patent a higher form of life. But among the arguments that were made to the court, a number of the arguments were that the mouse was mostly natural. It had only been modified in a small number of places. So you couldn't really claim the whole mouse just because it had been modified in a couple of places. But the holding of the case, the rule of the case was really that a higher organism could not be patented, even if it had been genetically modified. Now, 2 years later, the Supreme Court in Canada was asked a very similar question about genetically modified crops. And interestingly, the Supreme Court found that you also could not patent a genetically modified crop. But what it did say is that although you can't patent the entire organism, you might be able to patent individual cells of the organism, and you certainly could patent individual genes of the organism, especially if they were synthetic. Now, if you think about it logically, you would much rather be able to claim an organism based on their genes than based on the entire organism, because you get more scope if you're able to control their genes. So in a backwards sort of way, the Canadian Supreme Court reversed itself in the 2004 ruling in Monsanto versus Schmeiser and made it easy to patent synthetic genes. In the United States, in the meantime, this gene rush was running into problems. One of the problems is that a very popular form of patent was on what's called an expressed sequence tag. It's not a full gene, it's just a part of a gene. And in 2005, a very influential appeals court, it's the Patent Appeals Court, said that you cannot patent a fragment of a gene because it doesn't really do anything. This eventually led up to the case that we'll look at in a second, the Myriad case, but it started to cast doubt over whether the investment in patenting genes was a good investment by the biotech industry. Now, empirically, it seems like there's very, very few litigations that involve human genes, almost none, and there's no known finding of patent infringement of a human gene patent, which is interesting. It may signal that they're not lucrative, they're not useful, or that people are afraid to press their legal rights. In 2007, Xavier Becerra, who is now a member of President Biden's cabinet, he proposed something called the Genomic Research and Accessibility Act. And in this act, the idea was that it would be very, very hard to patent a gene, very hard to control a gene, whether it's synthetic or natural. And then in the America Invents Act, which is American statute that became law in 2012, the American Invents Act includes a provision that says you cannot patent a part of a human body. You can't patent an invention which is closely related to the human body. And many people have interpreted that to include human genes. Okay, so enter a civil rights organization called the American Civil Liberties Union, or the ACLU. They took up the case of several women who had had a genetic test to see whether they were predisposed to breast cancer. The test was for the BRCA1 and the BRCA2 genes, which can be an indicator of whether you are likely to develop breast cancer. They went, using their medical insurance, they went and got their tests, and the tests were positive. But rather than get radical surgery that could change their life on just the strength of one test, what they did was they asked if they could get a second test, and their insurance company said no. You can only get one test, you'll have to pay for it yourself. Well, the tests cost about $3,000 to $4,000 at the time. So the American Civil Liberties Union, they said, this is unfair. It's unfair to deny people information about themselves just because there's a patent on the BRCA1 and BRCA2 genes. So a couple of physicians decided they would offer these tests for free. These tests cost very little to carry out. And they said, we'll do the tests for you. But the company that owned the patents, Myriad, said, no, you can't, that's patent infringement. And eventually the ACLU decided to take this to court and to challenge the United States Patent and Trademark Office, which had been giving patents on human genes. And what the court ended up saying, with the first level court, the trial court, is that genes purification from the body using well-known techniques renders it patentable by transforming it into something distinctly different in character. Many, however, including scientists in the field of molecular biology and genomics, have considered this practice and this is very, very, very unkind, I think, a lawyer's trick that circumvents the prohibitions on the direct patenting of DNA in our bodies, but which in practice reaches the same result. And the court said, therefore, they are deemed unpatentable subject matter. So a little different, I think, than the European approach. At the subject matter level, these were deemed unpatentable. Well, in the meantime, there was another court case being decided by the Supreme Court. And this case is called Mayo versus Prometheus. And the Mayo versus Prometheus case was not on gene patents, it was on other aspects of biotech, but the Supreme Court said, you cannot patent some types of biotechnology, not genes, genes were not at issue, but it cast doubt on the patentability of a number of biotechnologies. Not only that, but the Supreme Court, which was about to hear the appeal in the previous case, the human gene case, which by this point was called AMP versus Myriad, the Supreme Court said that the appeals court should take a careful look at its decision in Mayo versus Prometheus, take that into account before making a decision. The appeals court did. They found that actually human genes were patentable, and the Supreme Court said, we don't think so. We're going to hear the appeal at the Supreme Court level. So they did. It went up to the Supreme Court, and an almost unprecedented amount of interest accompanied this case. So one of the things that happened was the Department of Justice, a branch of the US government, they filed a friend of the court brief which is very influential. And what they said was, there should be a difference between isolated but otherwise unmodified genomic DNA, or you might think of that as natural DNA, and human engineered DNA molecules, synthetic DNA. This marks a big, big difference in the official policy of the US government. And very interestingly, the Department of Justice did not even consult the Patent Office. They didn't tell the Patent Office they were changing their view. And in the meantime, the Patent Office had been allowing patents on natural source DNA. So 2013, the Supreme Court found that unmodified DNA was unpatentable. And the conclusion, this concluded sort of a long, long line of disputes about whether you could patent human DNA. But what was fascinating is that the decision explicitly said that synthetic DNA, modified DNA, was still patentable. I think a number of the speakers mentioned that there's this idea of unease with human-related patents. And I think that this played into these decisions. And I would direct your attention, if you're interested in this issue, to Radhika Rao, who talks about unease with the ownership of living bodies. Michelle Goodwin, who talks about the unease with dead bodies, including organs that might be transplanted. And then Ray Madoff, who has also written just in general about the unease with human property and human patent property. And there's also a number of American statutes that specifically deal with aspects of genetics like privacy. And those are the HIPAA and the GINA statutes. And I won't get into those. And then there's section 33 that I mentioned already that says that human-related inventions are not patentable. So we've already seen synthetic biology. The way that I think about it is that there's two levels of it. There's the redesign of existing things, and then there's the brand new de novo design of parts or entire organisms or cells. There's a number of historic milestones that I think are interesting. One is biological engineering was first coined in 1963. The first recombined DNA was made in 1973. The human genome was sequenced several times by 2000, 2003. And synthetic biology was successfully used to make a drug called artemisinin, which has therapeutic effects against malaria, as long ago as 2006. We've already talked about the synthesis of artificial organisms by Craig Venter's group. I want to point out as well, though, that an entire book on synthetic biology was actually made available in DNA. So you can get the print version if you want, or you can get the DNA version, and they will send you a copy of a long strings of A's, G's, T's, and C's that encode the entire book. Synthetic biology, as you've already heard, it encodes a number of sort of interesting goals and features. So one is to synthesize large DNA molecules, and another is to design circuits of these genes, integrate them with cells, and eventually make synthetic organisms, if possible. There's a consciousness on engineering approaches, which has already been mentioned. And very, very interestingly to me, a lot of the scientists in the field began their education as engineers and moved into biology because they thought that they could revolutionize the way that biology was done using engineering and computer science approaches. One of the ethos that came out of synthetic biology was the whole idea of democratizing it, of making it available not just to scientists, but to everybody, including to citizens, including to high school students. Now, whether this is a good or a bad idea, this is an ethos that was embedded in the movement of synthetic biology. And this is the cover of the top science magazine in the world, Nature. And if you get this issue, there's an entire comic book inside that explains synthetic biology. And the very idea of a comic book is democratizing. They wanted this to be accessible to everyone, the ideas in it. Now, I won't go over all these institutions, but I want to point out that in a short period of time, many institutions have arisen around synthetic biology in Europe, in the United States, and increasingly in Asia. Some of these are very influential, some of them less so, but all of these organizations are trying to establish norms and promote legal perspectives on synthetic biology. The one that I want to focus in on briefly is the Biobricks Foundation. Their mission's to promote responsible synthetic biology. They manage something called the Registry of Standard Biological Parts, and they run an Olympics every year that is attended by thousands of people, in which teams from around the world, including Italy, I checked, Italy has sent many teams. These teams come and they show off their genetically modified organisms using the techniques of synthetic biology, and they win medals. They win gold medals, silver medals, bronze medals. It's like the Olympics crossed with Lollapalooza. It's a big festival of synthetic biology, and it's deliberately meant to increase enthusiasm in this field. And one of the ethics is that open works better than proprietary, that you shouldn't patent things even if you can, because it's better to make these parts available to the world. I won't mention too much about the registry other than to say that it has a library of 20,000 Lego pieces, which are genes, 20,000 Lego genes. And these genes are the raw material that synthetic biologists who engage with this Olympic event use every year to make their organisms and cells and genetic circuits. And they're expected to give their parts back to the registry when they're finished. If they design new parts, they're supposed to give it back. And so the phrase is get some, give some. And this is why the registry has grown so fast. There's a specific format of these parts, and they're supposed to sort of biologically click together. They're standard, they have to have a unique sequence, they're identified, and you can use them to make all sorts of different things. Here's an example of a piece. It's called a LASR activator, but this is one of the Biobricks, and it's got the code up above where you could identify to it specifically, and they could send you this part. Now, a lot of standards have arisen around this community, including standards that have implications for patents. And again, I won't talk very much about this, but one of the interesting things is that standards is a feature of engineering and computer science, much less so of biology. And so synthetic biology has adopted the idea that standards are an important way to govern the community. And again, there's the ethos of open innovation, which has been baked into the idea of standards. There's technical standards. There are IP policies, patent policies, which try and encourage people engaged in synthetic biology to protect or not protect their synthetic biological inventions in particular ways. There are also software tools, and these software tools are meant to make it easier to identify, to analyze, and to build new synthetic biological tools. And there are also biosecurity standards, and biosecurity has already been mentioned, but one of the interesting things is that this has been the hardest area to get a standard in, biosecurity. It would seem to be the one that should frighten us the most, and yet it's the one that people can't agree on, that they can agree on the least. And this is sort of a summary of the different standards on biosecurity. So I'm almost done, Amedeo, don't worry, we're drawing to a close here, but I wanted to draw your attention to a license called the Open Source Biology License, which tries to explicitly deal with the ownership and the patenting of synthetic biology. It is in keeping with the open ethos, and the idea is to copy the licenses, not copy, but to use as a guide the licenses that deal with open source software. And this is essentially what the agreement does. It encourages people to adopt legal standards and ethical standards of use. It encourages people only to use the parts for responsible purposes. And it also encourages people to declare whether or not the parts that they have developed are covered by a patent or are not covered by a patent. Drew Endy, who is sort of the architect of this agreement, this contract, was named a champion of change by President Obama, in part for his leadership on this Biobricks public agreement. The agreement covers the iGEM competition to a certain degree. This is the Olympics event, and teams are supposed to give back their inventions to the registry and to keep them as free from patents as possible. Now, very briefly, I want to show you some interesting data about whether they give it back or not. So the Olympic events are participated in by people from the US, Europe, Asia, and other places. And you'll notice that participation has gone up quite a bit. And this is the use versus contribution. So black is the number of parts that they have used from the Registry of Standard Biological Parts, and white is the number of new parts that they have given back to the registry at the end of the Olympics. And what you can see is that there's an enthusiasm to give back new parts, not just to use what's there. And this sort of flies in the face, I think, of some economic ideas that there should be a tragedy of the commons, that people should only use and not contribute back. There's different types of projects, and these different types of projects have been growing at different rates. Therapeutic projects have been the fastest growing. And then there's all sorts of different types of parts as well. Now, final note that I will give, and then I'll cede the floor, is that there have been developments since 2012, which have, I think, changed the entire field of synthetic biology, accelerated it like pouring gasoline on a campfire. Prior regulation, I think, has been based on the relatively inefficient genetic engineering technologies that existed, but CRISPR-Cas9 and the other technologies that have come out of this, I think, have accelerated the field of synthetic biology onto a new trajectory. And these are exactly the kinds of inventions that, at least in the United States and Canada, appear to be highly patentable. So there are three different types. CRISPR-Cas9 is the foundation technology. And essentially it allows you to snip open DNA and then to modify the base pairs of DNA, and then the cell will seal it back together. There is a more advanced method called base editing, invented by David Liu, actually at the Broad Institute, where rather than snip the DNA apart, you actually chemically change the A's, G's, T's, and C's into other bases. You change them while they're part of DNA by changing their chemistry. And then more recently, also from the Broad Institute, has been something called prime editing, also by David Liu. Prime editing is a very versatile, a very precise, and a very accurate way of genome editing. And the estimates are that it should be usable to fix up to 89% of current human genetic diseases based on the structure of the mutations that cause those diseases. So this is basically what CRISPR-Cas9 looks like. This is what base editing looks like. This is what prime editing looks like. And the final little note, Amedeo, don't worry, I'm almost done, is I've been involved in a project out of Britain to consider how and if to bring back the great auk, which is an extinct bird. We actually visited the islands that the great auk used to live on, the Farne Islands, off the eastern shore of Northumberland in England. And the idea is that maybe with new genome technology, new synthetic technology, just like the mammoth, you might be able to bring back things like the dodo or the auk. And this is actually an auk egg that I was allowed to hold. I didn't drop it, I didn't break it. So the conclusions: rapid advance, lots of patenting in North America of synthetic DNA and synthetic inventions. Open biology is a real alternative and is being pushed hard by people in the field. And there's many patent applications, but even more legal and ethical challenges that remain. And so that's it, Amedeo.
Oh, thank you so much, Andrew, for this fascinating presentation. And now we move rapidly to the presentation by Marina Tavassi. It's a pleasure to introduce Marina Tavassi. We worked together in the Court of Appeal of Milan. She was the president of the court. I was the president of a chamber of the court. And now we are together professor at the University of Pavia. And it is nice, again, she is a real authority in the field of patents and law in Italy and in Europe as well. And she had several occasions to give her contribution to the European elaboration of legal trends and standards. Marina, you have the floor, please. No, we can't hear.
Sorry. Thank you. Thank you very much, Amedeo, for this presentation that I am very happy to be here and to be part of this very, very interesting presentation made by the so brilliant speakers. I think that we all have learned a lot today, and we spent 2 hours, but very, very interesting opportunities for all of us, the students, the professors, and all the audience. The reports we heard dealt with very complex issues, but the ability of the speakers has expressed the issues in such a way as to make us understand a very difficult concept in an extremely clear way. But I think that remain open a lot of difficult questions, a lot of problems to which is very difficult to give an answer. But this is not also for us today, but for the entire society and for the expert scientists too. Our brilliant speakers dealt with synthetic biology, a promising new biotechnology that has generated a lot of scientific, social, ethical, and political interest. However, issues concerning the patentability of synthetic biology inventions remains open, even if we have examined in detail every possible solution, alternative, criticality of every single choice. It remains uncertain whether these inventions are patentable on the basis of existing law and case law, at least in Europe. As Ilaria, Delisa, and Andrew Torrance reports have addressed this question, focusing specifically on the suitability of the topic and moral criteria, and on the acceptability of the invention, and all the other difficulties that arisen from these that are a reason for this new scientific approach. They have provided us an overview of the legislation and, above all, Andrew, of the decision that are apply to synbio patents, and examine this new technology in light of the ongoing debate on the patentability of biotechnology and possible alternatives in general in the US and Europe systems. All of the today's speakers have offered the scientific and the legal analysis on the problem, complemented by the practical examination of several possible applications submitted to the European and US system, but it is very, very difficult to find a general solution. It is interesting to note that the European Directive 44 dates back 23 years ago, is the Directive 44 of 1998 regarding the biotechnology. The European legislator has intervened to regulate the biotechnology sector, considering that biotechnology and genetic engineering are playing an increasingly important role 23 years ago in a broad range of industries. In that period, the protection of biotechnological invention was certainly of fundamental importance, not only for the European community's industrial development, but also to answer to many questions of an ethical and social nature that those problems and the progress of science posed. The Italian Industrial Property Code is fully harmonized with the Biotech Directive. The entire section 4 bis, article 81 bis to 81 octies was inserted by legislative decree number 131 of August 2010, precisely to transpose the European directive into national law. The text of Article 81 ter reproduces the definition of biological material and process contained in the directive. I have to say that in my experience and in the experience of one of the most specialized department, the specialized section for enterprises of the Tribunal and of the Court of Appeal of Milan, we don't have cases regarding the application of these rules. Very, very rare situation presented the opportunities to deal with these articles and these rules. The articles, the Italian articles on biotechnology repropose, in compliance with the peculiarity of the subject, the requirement for patentability provided in general by Article 46 regarding novelty, 48 regarding inventive activity, 49 industrial applicability, 50, the lawfulness, 51, the sufficient description, as for the patent in general in our industrial property code, even if the wording of Article 81 quater is more detailed and focused on the specific topic of biotechnology. The same happens for Article 81 quinques, which deals with exclusions from the possibility of registering as a patent. The hypothesis reproduced the more generic provision of Article 45, paragraph 4, which is expressly referred to in paragraph 1 of Article 81 quinques, but the wording is more specific, with 3 provisions and 5 subcategories precisely refer to the peculiarity of the biotechnology. The closing rule that provides in any event, any technical process which uses human embryonic cells may not be patented is extremely significant and very decisive in its all-encompassing nature with regard to the human embryonic, and is also important to underline as a possible reference also for to give an answer to the problem of today. We know because Professor Delisa recalled us Article 27 of the TRIPS agreement that is the basis for for the solution of this particular problem. And in my opinion, it is very important to remember the paragraph 3 of this article that suggests to the member state that it's possible also exclude from patentability diagnosis, therapeutic and surgical methods for the treatment of humans or animals, plants and animals other than microorganisms, and essentially biological processes for the production of plant or animals other than non-biological and microbiological processes. However, continues the paragraph 3 of such rule, members shall provide for the protection of plant varieties, either by patents or by an effective sui generis system, or by any combination thereof. And I think that our problem of today deserves to receive a sui generis answer, because we have heard today what are the differences between other field of the technological invention and the possible technological patents. I would like also to remember the last suggestion made by the European Parliament regarding a possible close problem that is close with the development of the research in synthetic biology, and is the field of the artificial intelligence. It is worth mentioning that the European Parliament has recently, on October 2020, adopted 3 resolutions containing recommendations for the European Commission in view of the drafting of future regulation on artificial intelligence, a topic very close to the one we are dealing with today. The resolutions contain indication aims at defining a European legal framework of civil liability, ethical principle, and the protection of intellectual property rights in artificial intelligence systems. Those resolutions regard civil liability regime, regard ethical aspects of artificial intelligence, related technology, and on intellectual property rights for the development of artificial intelligence technologies. But in my opinion, so reading this long resolution, they are very, very generic. And in my opinion, this is the time in which is necessary to pass from the generic assertion to the specific rules of this sector, because even if we have specialized judges in Italy and in Europe, and we have the umbrella of the European Court of Justice, it's not possible in this very sensitive field to leave judges deciding alone the necessary step in order to make order in this particular field. And I remember that Andrew denounced the possible assault on gene patents. And this is also a risk, because we know that the protection resulting from the granting of a patent gives the holder a micro monopoly position on a particular technological solution that, in my opinion, is very dangerous in this field regarding all the possible implementation. This application could be very positive, but also negative, and is very important to have a certain regulation of this specific field. Thank you for your attention. We pass our times, and sorry for that. Thanks again, because also on behalf of our students, Amedeo, because I think that it has been a very, very interesting opportunities for all of us.
Sorry. Thank you so much, Marina. And before giving the floor to all the speakers for just one sentence or comment, whatever they want, because we have some minutes for a little discussion. I would just add to what Marina said about the European Parliament. Just yesterday, there was an informal information of a draft resolution by the European Commission of a regulation on artificial intelligence that would encompass also these aspects of the applications in the field of biology. And as far as I know, the criterion adopted by the European Commission is the criterion of high-risk technology. And perhaps it's possible to consider this kind of scientific applications, or at least some of them, they can be considered clearly high risk. High risk is from the point of view, of course, also of the society and so on. But this is a work in progress. So let's go to in the order, however you want. Silvia, do you want to make a comment?
Yeah, well, I think I've learned a lot. I don't know if I've understood everything from the law side, of course, but what I think is that the frontiers in science can be, cannot be, how to say, jailed. Science cannot be jailed in any way. So I think science has to go on, and also why not to produce new organism, maybe coming from the past. I don't think it would not be so easy, telling the truth, but why not? Maybe not in 10 years, maybe in 50 years, but why not? What is really important is the framework in the society in which these results are posted. I have a good feeling for open biology. Because I think that an open knowledge is really helpful. The best example is Wikipedia from another side of possible knowledge. But in fact, it's not so difficult to manage the genome, and in fact, it's not so difficult to manage cells. So I don't know. I rely on you all.
Thank you, Silvia. And Lorenzo, what about you?
Yeah, I share some of the thoughts of Silvia. So about the importance of the open source, open access parts and things that can be done. I found myself very much in the description made by Professor Torrance because I started everything with the iGEM competition many years ago. So I started with that because I was an engineer doing my master thesis, and so I didn't know nearly anything about practical working on the bench with biology, and so it was really useful for me to have an archive that was shipped on my desk, and it had not so many parts not as many parts are they are now, but like 800 or something like that. It was there, and we still use that in the lab right now to build up things. Because they include a lot of things. If they are high quality, they work, and why not? We are also working on the same gene that he showed in the slides, so it's more or less that's why I found it very useful. And so like me, since I found my early career in that role, I think that many others can benefit from the same things. As he said, it is a different things from patenting, because some of the parts can be patented and so on. But the openness of the community is very important to make scientists grow, because otherwise. Probably on my last sentence of the presentation, one of the last sentences, if one of the milestones in the last years that is a really big achievement of synthetic biology is that now biology has been accessible to many people not really biologists. You don't have to work to know everything, but you can hide the complexity of many things if you think about modules and Biobricks parts and so on. And it is quite simplifying as a designer. And then the rest of the biology is essential to provide us the right toolkit of parts, because otherwise we don't have nothing to work on. That's the main point. I don't have nothing about the low part. It has been very useful to me, but yeah, I'm not the right person to comment on that. So thank you.
Okay, thank you, Lorenzo. Ilaria?
Well, I just wanted to touch upon what they said. And first of all, it was really interesting. I learned so much about the topic, both scientific and legally. So really, thank you very much also from my side. I would say the issue of openness or patenting, well, from a lawyer's perspective, I think more realistically, I think the bigger the companies involved and also people who have a strong tradition with patenting. I mean, we have people like Craig Venter who participate in the human genome process, who has somehow a track record for strong patenting. So I think that might be kind of a clash of systems. But what I found most interesting, and this I agree 100% with Professor Tavassi, is that also the legal system also needs to wake up to those new technologies, because clearly, as Professor Garagna said, they're not stopping. Science is not stopping. It's developing something new. And just expecting judges and lawyers to have that kind of scientific feeling for what is correct, what could be issues, how to deal with those very complex points, it's a recipe for trouble, I find, because then in 10 years from now, things will land on the table of judges. And I wouldn't say then it's too late, but we don't have the American system when then a Supreme Court can take years to get there, but afterwards they can somehow give a different input. We have a EU directive that was first drafted in 1988, so it's now turned more than 30, and it's quite an older framework. So I also wish that this would get updated and that the legislation will catch up with what is out there.
Okay, thank you, Ilaria. Andrew?
Well, I hope this doesn't count as my sentence, but I just want to say it's been an honor being part of this wonderful panel. As I said before, I learned so much. What I want to say is that there's a phrase that Charles Darwin used that I think it's provocative, but it may go to what Ilaria just said. And I'll mention my son's name is Darwin too. Professor Santosuosso knows, he's met my son Darwin. So I'm a big fan of Darwin, but Darwin once said that, about Darwin, Theodore Dobzhansky once said, nothing in biology makes sense except in the light of evolution. I think nothing in the legal regulation of synthetic biology makes sense except in the light of biology. And I think that a number of speakers have already mentioned this. I think we need to weave together the science and the law more closely than we've done in the past, so that the law can meaningfully grapple with the issues and not just settle disputes that have already occurred, but also set the course for the future. Allow science to be as free as it can be without creating the societal problems that Marina, I think, mentioned, that there are these problems that could arise. So I think that it's an integration that is necessary, and that we need to bear in mind biology when we're thinking about the law that pertains to synthetic biology.
Thank you, Andrew. Marina?
I completely agree. This is a very technical field in which it's necessary to have an interaction among technologists and lawyer, also because the technology ran away in a very rapid sense, and it's very difficult to maintain updated our rules and our approach, because I have to be sincere, a lot of things that we have heard today are completely new, are completely surprising, sometimes so also shocking for a number of us. And I think that for the legislator, for the European legislators, it's very difficult to construct a system of rules that is enough general to cover all the possible steps that the technology and the research can reach in a short period. And this is the very important challenge for our legislator.
Well, just one word to finish. I thank you all for your wonderful contributions and for staying together. And I am not surprised of the need of a dialogue between science and law, because it is our mission. In the last 20 years, we have worked hardly with Silvia Garagna and other people at the Center ECLT, but it is always a new challenge, and it is our pleasure to be in this challenge. But I have a good news. The good news is that the University of Pavia has recently approved to have again Professor Torrance as a visiting professor the next academic year in Pavia. And we really hope to have him in person in order to organize, okay, synthetic biology and law, and year later, what news? It can become a date every year if we want, of course, but it is our pleasure to have this kind of discussion. I thank you all again. I thank the ECLT Center and the IUSS Pavia for hosting us, and let's keep in touch. Best.
Thank you very much.
The best.
Bye.
Thank you.
Bye. Thank you.
Ciao.
Ciao.